Since cell cycle and cell biology is one of the most fundamental things in biology, I decided to make this video for class 11th as well as for higher studies. So wherever I am going out of the NCERT, I am going to star mark that area, and you can also refer to the description for timestamps. In the previous video, we went on a trip to understand the overview of the cell cycle, and we did the interphase in detail. If you haven't watched that video, I'll link it in the description. You can refer to it and then come back here to continue. Mitosis is the type of division where the number of chromosomes in the parent is same as the number of chromosomes in the progeny. So if the parent is diploid, the progeny cells are also going to be diploid. And this is the reason why mitosis is also known as equational division. Mitosis in itself is a continuous process, but for our understanding, we divide it into 4 different phases. Now these 4 phases can be remembered by a word PMAT. You can either imagine a dog peeing on a mat, or in itself this word is catchy enough that you repeat it in your head a few times, you will be able to remember it. How PMAT helps is that the 4 phases are the prophase, the metaphase, anaphase, and finally the telophase. At the end of these 4 phases, the nucleus of a cell divides into 2. The division of the nucleus of the cell is known as karyokinesis. And as one cell is formed with 2 daughter nuclei, the cytoplasm of the cell divides, and this results in formation of 2 daughter cells. The division of the cytoplasm is known as cytokinesis. This karyokinesis and cytokinesis eventually lead to formation of different daughter cells. Now one situation can be where the karyokinesis is immediately followed by the division of the cytoplasm. This will lead to formation of 2 cells with 1 nuclei or uninucleate cells. There can also be a situation where the division of the nucleus is not immediately followed by the division of the cytoplasm. This will lead to formation of a cell with more than 1 nuclei or multinucleate cell. This condition is known as syncytium. And a very good example of this condition is the liquid part of a coconut. In there, the division of the nuclei is not followed by the division of the cell, and therefore there are many free floating nuclei present in the liquid endosperm of the coconut. As we start to understand mitosis in detail, the first phase that we start with is the prophase. The prophase of a mitotic cell division is marked by the condensation of the chromosomes. Why the genetic material needs to be condensed is because it is present in a thread-like condition which occupies a lot of space so as to make the transcription of proteins easier. But once we have to divide this genetic material, it is always easier to condense it in a very small form and divide it into the 2 daughter cells, and it can unwind again. This is something like you packing up your papers into files, those files into some cabinets, and those cabinets in a room. As you start working with your papers, again, you will know where everything is present, and it will be easier for you to divide it if you have to. During the S phase, all the genetic material that is present duplicates itself. And once it does, there are 2 copies of every single chromosome that is present. These 2 copies are then held together at the centromere, and these copies are known as sister chromatids, and each one of them is a chromatid. Along with this centromere, there is another disc-shaped structure present around this chromosome, and this is known as a kinetochore. These sister chromatids are held together with the help of a protein, and this protein is known as cohesin protein. This cohesin protein is then degenerated eventually to separate these two chromatids. Since the prophase is the continuing process, its initiation is marked by starting of condensation of the genetic material. As the genetic material condenses, there are several other changes that take place in the cell, and therefore the end of the prophase is marked by several events. One of them is this completion of the condensation. Along with this, during the prophase, the nuclear envelope and all the organelles start to degenerate in the cell. So the end is marked by disappearance of organelles and the nuclear envelope. As you can see in the diagram, I have represented the degeneration with this dotted line. Another important thing that you can notice here are these red lines. These red lines are the centrioles of the cell. The centrioles are also duplicated in the S phase, and these centrioles then start to form a spindle-like structure. The two centrioles are present on the opposite ends of the cell, and they give rise to a mitotic spindle. This spindle is formed with the help of microtubules. These microtubules are of three different types. The first one are the kinetochore microtubules. These attach to the kinetochores that are present, the disc-shaped structures that are present on the two opposite ends of a chromosome. These have chromosomal interactions. The second type of microtubules present are polar microtubules. These do not have any chromosomal interactions but are just present adjacent to each other. And the third and final type are astral microtubules. These are not even directed towards the chromosome but towards the cortex of the cell. And the three types of microtubules are all polar. That is, the one end that is embedded into the pole is negative, and the end that is towards the chromosome or away from the pole is positive. So this structure is known as the mitotic spindle, and this is the structure through which the two different sister chromatids are separated from each other. As we move on from the prophase, we enter the metaphase. The metaphase process has an M, and according to our PMAT, the M will represent middle, which means that in this phase, all the chromosomes present in the cell will align in the middle. The metaphase initiation is marked by complete disappearance of the nuclear envelope. And this is the phase where the chromosomes are best observed. In this phase, the microtubules start to join with the kinetochores present on the chromosomes and therefore align them along the middle of the spindle. and this is known as the metaphase plate. The kinetochores function on both the directions. This is the law of Newton. For every reaction, there will be an equal and opposite reaction. As these microtubules attach to the chromosomes in one direction, that is, as they come along and attach to one kinetochore, they will oscillate for a while and then become stable as the second one from the other direction attaches itself. As these chromosomes attach to this microtubule, there will be a force that will eject them towards the middle, and there will also be a force that will draw them towards this pole of these microtubules. And this force is known as polar ejection force. These two forces combine to align these chromosomes in the middle on the metaphase plate. And this entire process of one microtubule from one end coming in and searching for the chromosome and attaching to it, and then waiting for the other one from the other side to come and attach as well, and then this oscillation due to this polar ejection force and the force which will align them in the middle, this entire process is known as congression. As the chromosomes are aligned in the middle, they will now start moving towards the two opposite poles along with the microtubules with the help of their kinetochores. This movement occurs in the phase Anaphase. And as this has an A, you can remember it by away. The sister chromatids start moving away from each other to the two opposite poles. And this movement is possible because the kinetochores start to shorten, and there's also a pulling force that is possible because of these astral microtubules that will pull these sister chromatids to the opposite poles. So there's a push force because of the shortening of the microtubules, the kinetochore microtubules to be precise, and there's a pulling force from the poles. At the end of the anaphase, these chromosomes reach the two opposite ends, and that marks the starting of the final phase or the telophase. The final phase or the telophase is the situation where there are two nuclei present now. The chromatids have reached the two opposite poles, and everything that had disappeared is now starting to form again. The daughter chromatids arrive at the poles. The mitotic spindle that had formed now disappears. The nuclear envelope that I have represented here with dotted lines, it starts to reappear along with the nucleolus that is present in the cell. And one very important thing is that all the chromatids that had first condensed to form chromosomes now start to expand again. This is the final step in the mitotic division or the division of the nucleus of a cell. After the nucleus divides, the next step is the cytokinesis or the step where the cytoplasm of the cell divides to form two daughter cells. Cytokinesis is a pretty long and stepwise process, but in a nutshell, it occurs in two different ways in animals and plants. In the animals, there is no cell wall surrounding the cell. So, it starts with formation of a furrow in the cytoplasm. And as this furrow grows, the cell eventually divides into two cells. However, in the plant cell, there is a very hard cell wall surrounding the cell. So, there cannot be a presence of a furrow in the plant cell. Therefore, the cytokinesis in a plant starts from the middle and then it moves towards the ends. The cytokinesis starts with formation of a cell plate. This cell plate grows to the opposite poles and eventually divides this cell into two parts. This cell plate later forms the middle lamella between the two cells. Along with the genetic material, all the organelles that are present in a cell are also equally divided. Now we are going to look into the entire process of cytokinesis in detail in the future videos. For now, this is the basic difference between the animal and the plant cytokinesis. One thing that we need to know now is why exactly mitosis is needed in the cell. Why must a cell expend so much of its energy in dividing, and what are the significances of mitosis? Significances of mitosis are several. One of the most obvious and the important ones is the genetically identical daughter cells production. The parent and the progeny have same number of chromosomes and the same genetic material. So, this plays a role in formation of same types of a large number of cells. And this feature of mitosis helps in the growth in the multicellular organisms. Our development, our wound healing, and the growth in size and height of an organism occurs because of this mitotic division. Another important thing that happens in the cell is that eventually it starts growing in size. And once a cell reaches a situation where the cytoplasmic content is very high than the nuclear content, the cell divides. So mitosis also helps in restoring that nucleocytoplasmic ratio of a cell. Along with this, mitosis helps in cell repair, which is basically wound healing and even taking care of our internal organs and protecting it from all of the damages. A damaged cell can easily be replaced by the process of mitosis. And in meristematic tissues of a plant, mitosis leads to continual growth throughout the life. So this is the entire process of mitosis. First, the parent cell had its genetic material duplicated, and that entire genetic material was then distributed equally among the two daughter cells. I hope you liked the video. If you did, don't forget to press the like button and subscribe to my channel to get all the future updates of the next part of the cell cycle as well as other topics of biology. Thanks for watching.